Okay, part 3. This should be the last video. So essentially what I'm going to do now is I'm going to highlight in words what we saw in the figure, the previous slide. And I'm also going to talk a little bit more about those portions of the pathway. All right, so aerobic respiration, you're always going to think it requires oxygen, like on demand all the time. So what we're doing in aerobic respiration is we're taking that glucose that we obtained from digestion, and we're going to use it in the presence of the oxygen, and we're essentially going to make some byproducts. We're going to make carbon dioxide. We're also going to make water. Okay, but we also know that we need to get that energy out from our catabolism of glucose, and that's going to be in the form of ATP. So we're usually thinking 36 to 38 ATPs. We will go through all of those 3 steps: glycolysis, Krebs cycle, and electron transport chain. Remember, the end result then is that free oxygen is going to be the final electron and hydrogen acceptor in this pathway. So characteristic of many bacteria, fungi, protozoa, and animals, absolutely. Aerobic respiration changed the life of organisms on the planet because they could get so much more energy. They could do so much more work on the amount of effort of acquiring nutrients. So that may not make a lot of sense to you, but if you were living off of anaerobic respiration or fermentation, 2 ATPs, you literally would not be able to get anything done in a day because you wouldn't have enough energy for your cells. Okay. The overall equation for aerobic cellular respiration is at the bottom of the slide. It is also on your worksheet for the week. And I've named each of the components. Remember, on the left-hand side of the equation, we have our reactants. So glucose, C6H12O6, and oxygen, which is O2. We have that arrow. Of course, there's going to be enzymes related to the arrow. But on the right-hand side, we have our products. So CO2, or carbon dioxide, and then water. So this is a balanced equation. The ATP, while it doesn't belong anywhere in our balanced equation, we do know that that process is going to yield the 38 ATP, so I put it there. Okay, anaerobic respiration, anaerobes. And when you put AN on a word, it negates it. So instead of it being high oxygen, we're thinking low oxygen. So these are going to be with or without oxygen. And essentially, we're still going to go through glycolysis, Krebs cycle, and the electron transport chain. But instead of using oxygen, we're going to use other oxygen-containing compounds as the final terminal electron acceptor. So you see 3 there, and I also have a table with it as well. So we will come back to these the names for these chemicals in just a bit. And then the last one was fermentation. So complete oxidation of glucose, but no oxygen is required. Oxidation essentially means we're going to go through those processes, but we're just going to go through glycolysis. Remember, no Krebs cycle, no electron transport chain, and some other organic compound is going to be the final receptor. It is not going to be anything with oxygen in it, which kind of makes sense. Low oxygen, low ATP. Okay, first part of these metabolic pathways is glycolysis, which is called glycolysis. Glyco is referring to the glucose, and lysis means to split. So that's why in this figure it looks like a tuning fork, because you got the base of the tuning fork and then you have the tuning rods here. So it's branched because we take a 6-carbon glucose molecule, and the very first thing we're going to do is split it into 3 carbon compounds. All right, so that's what glycolysis means. We are going to make a little energy, and we're also going to make that NADH. I don't really want to talk about all those, but just essentially know that there are some ancillary cofactors and coenzymes that are being made that also are going to be recycled in our metabolic pathways. Okay, I don't need you to memorize all this stuff on the left-hand side. Just would like you to see here's glucose, and we are going to break it down into 3 carbon compounds. It happens to be called pyruvate. Okay, so you can see there, you can believe it. Okay, glycolysis literally means breaking down of glucose or splitting of glucose. All right, cycle. Here is our cyclic type of metabolic pathway. And what we see, if you want to look at it, we take one of those pyruvates and we put it into our Krebs cycle. It is a 3-carbon compound, but right at first, it is going to join with a 2-carbon compound and then something else, and it becomes essentially a 6-carbon compound. We're going to take out the CO2, so this is where our CO2 is coming from. In our chemical equation, we're going to take out some ATP. We're going to make some other stuff that's going to get recycled or used. We're going to crank out some more CO2, and by taking off 2 CO2s here, then we're going to reduce our 6 carbons to 4 carbons. And then there's some other steps, and then we get back to the acetyl-CoA. But the story is, it's a cycle, okay? 6, 4, recycle, recycle, recycle. Take off the carbons, so that's how we go from 6 to 4. So that's generally what's going on with the Krebs cycle. All right, the respiratory chain, which they're calling it, or the electron transport chain, is essentially happening. This is the last part of carbohydrate metabolism, carbohydrate catabolism for both aerobes and anaerobes. And what is going on here is essentially we are using some products. So don't really want to go into it, right? But here's something we saw before being generated in both the first 2 steps of respiration. And essentially what we're doing is we're generating a buildup of hydrogen protons on one side of this membrane. And then what happens then is those hydrogen protons move across or are acted upon by an enzyme called ATP synthase. And ATP synthase then is going to catalyze the reaction of ADP to ATP. So these hydrogen protons are producing a gradient where they're in higher concentration on this side of the membrane, and then that prompts them to essentially react with the ATP synthase, this enzyme, and put that high energy phosphate bond on the ADP to make the ATP. Then we have extra hydrogen protons, and so we're talking about, well, who's going to accept it? And so we see here on the left-hand side, oxygen is going to be the final acceptor, and we make water as a result. So that's why we see we have water as one of the products in cellular respiration. And then we see in anaerobic respiration, instead of it being oxygen, it's going to be some other oxygen-containing compound that is going to be the final electron acceptor from those hydrogen protons. Pause here if you want to read this and understand it a little bit differently, but I'm not going to read over the slide. And this one as well. Okay, so let's go back. Now we want to take a look at the final step of each of these results of carbohydrate catabolism. So the final step in aerobic respiration, as we said earlier, is going to be that the final electron acceptor is going to be accepted by oxygen, but there's a potential side reaction that can happen when there's an incomplete reduction of the oxygen. So essentially what we have is the production of what we call superoxide. So you see there is a net negative charge. So that's what we're calling it, superoxide. There's nothing joining that. And then we have the production of hydrogen peroxide. Well, hydrogen peroxide is something that has to be dealt with in cells, whether it's human cells or bacterial cells. So our aerobic organisms, thinking about those aerobic bacteria, have neutralizing enzymes that are going to essentially detoxify hydrogen peroxide. So we have superoxide dismutase and catalase, which are very good at converting hydrogen peroxide back to water and oxygen and not have this superoxide ion hanging around anymore. The last point, just giving us an example of a microorganism. So Streptococcus, again, we're thinking about a strand of coccus cells, and they lack the enzymes to break down those superoxide ions, but you know what? They still grow well on oxygen due to the production of an enzyme called peroxidase. So as I mentioned in the earlier part of our lecture, one of the examples I was using was peroxidase as an enzyme that breaks down hydrogen peroxide. So it turns out we have that as well. Potatoes have that. Anyway, it's super important to be able to detoxify the cell. Okay, anaerobic respiration. Final step, what's going on? So I told you before that the final electron acceptor is going to be an oxygen-containing compound. So we have nitric oxide, or NO, nitrous oxide, N2O, and even nitrogen gas. So some species, as it mentioned, Pseudomonas and Bacillus, possess enzymes that can further reduce nitrate to these various nitrogen oxygen-containing compounds. Now, we think about nitrogen gas in the atmosphere. We know that what we call denitrification is important to recycling nitrogen in the biosphere. If you've ever had a fish tank, then you probably know a lot about how to deal with nitrates and nitrites in your fish tank, but bacteria are very, very good at that, to reduce those into some pretty non-hazardous chemicals in your fish tank. Other oxygen-containing nutrients reduced anaerobically by various bacteria are the carbonates and sulfates. And we can see none of the anaerobic pathways produce as much ATP as the aerobic respiration pathway. Okay, so here's these other kind of a chart to some of the words I'd mentioned before. An oxygen-containing final electron acceptor. So NO3 is nitrate, SO4 sulfate, CO3 carbonate. And then we also see that there can be some products that are made as a result: nitrite, hydrogen sulfide, and methane. So these are some things, some products that we may actually use in an industrial process. So we use the bacteria to make these chemicals for us, or we might use microorganisms to help us break down some waste products. For example, if I'm at like a cheese plant or a Campbell's soup plant and I'm trying to degrade away some of the natural byproducts of our soup making or whatever, I might use microorganisms to do that and make other chemicals that can be purified and sold. Okay, the very last one here is fermentation. So let's take a look. So in fermentation, you see that we are essentially going to take that a 3-carbon compound. So remember how we took glucose and we split it into 2. Okay, so glycolysis happens, with fermentation. We have a 3-carbon compound then that can become one of 2 things. It's not going to go into the Krebs cycle, so it's either going to be converted to ultimately ethyl alcohol, or it's going to be converted to lactic acid. And again, you see some of these things we saw before, the NADH. What's interesting here is you see that the NADH lost its hydrogen here and now became NAD. We also see that we make some CO2. So if you know much about fermentation, you know that you have to, for example, if you're making wine or beer, you have to use a fermentation lock, which goes into a stopper. It has some water in it and a cap on it. It allows gases to move in and out. But essentially, no oxygen can get into your wine or your grape juice or your wort if you're making beer. And essentially, we need to allow CO2 to escape from whatever jug or bottle you're housing your wine or beer. So the CO2 has to escape. So we know that fermentation definitely is making CO2, and we definitely know that we're not getting as much ATP as a result. Okay, the very last, second to last slide, products of fermentation of microorganisms, of course, alcoholic beverages, of course, CO2. So if you like carbonated water or club soda, we can get that CO2 from the fermentation process, so we can capture that. Solvents like acetone and butanol actually comes from fermentation. Some organic acids, remember how I mentioned lactic acid in your muscles? Vitamins, antibiotics, and hormones. And then, sure, we can use microorganisms on a large scale to produce all kinds of various chemicals that we need. And it's mentioned in here, antibiotics, hormones, vitamins, and amino acid production. Oh, and that's the last slide. Okay, guys, thanks for hanging in. I'll see you next week. Be good.